So as we mentioned before, when you have an exergonic reaction, you can use it to drive an endergonic reaction that otherwise would not occur. And this is something that happens all the time in your metabolism. And here's an example. You need to make this amino acid, glutamine. And what glutamine is a glutamic acid with an ammonia group attached to its side chain. That's basically what it is. So again, this is one of the 20 amino acids that you have in your proteins, and without this glutamine you would not be able to make all the proteins necessary to survive, and you would have a very debilitating disease. So you have to do this, you have to make glutamine, but here's the problem that your metabolism faces. In order to make glutamine, what you do is you take glutamic acid, add the ammonia, but the delta G for that is positive 3.4 kilocalories per mole, which means for every mole of glutamine that you make, you have to add 3.4 kilocalories. Okay, so on the exam, I can just show you this and say, is this spontaneous or not spontaneous? And you should answer, it is not spontaneous because the delta G is positive. Is it exergonic or endergonic? Again, you should answer it's endergonic because, again, all three of those things relate. Whenever you have something that is a delta G positive, that's also not spontaneous and it's also endergonic. So this is an endergonic reaction. Your body cannot produce it without an outside energy source. So, to make glutamine, it isn't this simple. What your body does is this. First, it takes a glutamic acid and an ATP and takes off the gamma phosphate of the ATP and puts it on the glutamic acid to make glutamic acid phosphate. Now, remember, go back to, if you need to, go back to one of the earlier slides where we were looking at the ATP and we saw those 3 phosphates sticking off. I'm going to take a third of those phosphates, the gamma phosphate, put it on the glutamic acid to make glutamic acid phosphate. I'm left then, you should see in your mind, an ADP. Okay? Now, once I have the glutamic acid phosphate, that is not the same as glutamic acid. That's got more energy in it, a lot more energy. The energy was transferred from the ATP onto glutamic acid phosphate. So glutamic acid phosphate can then be used to make glutamine by attaching a ammonia group to it, but the phosphate then comes off. And now I've got my glutamine and the ADP and the free phosphate. Okay, now if we're looking at these reactions, and we're looking at them in terms of biochemistry, this is sufficient. But from physics, is this even possible? The only way that this is possible is if the delta G of this whole reaction system here is negative. Remember, things can't happen in this universe unless the delta G overall is negative. So how do we look at that to figure that out? Well, it's pretty straightforward, but you can't look at it this way. What you have to do is see 2 different reactions. The first reaction that you see here is this one. Glutamic acid plus ammonia makes glutamine. The delta G is plus 3.4 kilocalories. All right, so there's your glutamic acid, there's your ammonia, there's your glutamine. Okay, but then what's the other reaction? Well, the other reaction is everything that's left over. We looked at this, and this, and this, and now what's left over? Well, notice this disappears. This was formed and then disappears, so we can ignore it. And then what's left over is only the ATP, ADP, and phosphate. And you can see the ATP goes to ADP and free phosphate. So from physics, what we would see is this. This glutamic acid plus ammonia gives us glutamine. The delta G is plus 3.4 kilocalories per mole. Now, how much energy is released by ATP forming ADP and phosphate? Well, we know that already. That's minus 7.3 kilocalories per mole. Therefore, the total delta G of this reaction system is the delta G of these 2 reactions occurring simultaneously, which is just simply their addition. So the delta G is plus 3.4 kilocalories per mole minus 7.3 kilocalories per mole. Therefore, we end up with a number of 3.9 minus 3.9 kilocalories per mole. Now, is this spontaneous or not? Well, again, delta G is negative, so it's spontaneous, it's exergonic, and can occur. So this is something that can occur, and therefore your body can actually use that in this universe to create glutamine, and that's exactly how it does it. Here's another example from your actual metabolism. Suppose you take sucrose, table sugar, and put it into your coffee. And here is the structure of sucrose. Remember, it's a disaccharide. This side here is a glucose, and this here is a fructose. Fructose and glucose are isomers of each other, and therefore they have the exact same molecular formula but different structural formulas. And they're connected by this glycosidic linkage with the oxygen right here. Now, if you put that in your coffee, it then sweetens your coffee. Your tongue can detect it, and you can taste it as sweet. But what happens to it? Well, it goes into your stomach, not much. It goes into your small intestine, however, and then this happens. This sucrose then gets broken down into this, glucose and fructose. And it requires water to do that because the glycosidic linkage has to break. One of the hydrogens goes to the oxygen to form this OH group on this carbon. And the OH, the hydroxide group, hydroxyl group goes to this carbon to form this OH here. And that happens only in your small intestine. It doesn't happen in your coffee. It doesn't happen in your mouth. It doesn't happen in your stomach. It only happens in your small intestine. Okay, this reaction has a delta G that's less than 0. So again, is it spontaneous or not? Delta G is less than 0. It's spontaneous. It's exergonic. It releases energy. So this literally will release heat in your body. But the question is this: if it's spontaneous, then why doesn't this happen in your coffee? And why doesn't it happen in your mouth or your stomach? In fact, why doesn't it happen on the shelf? You got a big bag full of sucrose that you buy at the store. Why isn't it being broken down into glucose and fructose? Well, the answer is it is. It actually is being broken down into glucose and fructose. It is happening, but it's happening extremely slowly. This reaction is spontaneous, but it requires energy to get it started. That's a concept then that we need to deal with here, because your metabolism has to deal with that too. This reaction occurs, but it occurs so slowly that it isn't going to happen. If you buy a bag of this stuff, of this sugar, even if you put it in water and let it sit for the length of the time the universe is around, then maybe you'll be able to get it into glucose and fructose. But this reaction happens so slowly that in your time scale, just a space of weeks or days or months, it's not going to change very much at all for you to notice it. Okay, so there's something going on here. There's something happening, though, when it gets into the small intestine, that suddenly it happens much more rapidly. It occurs much, much faster. And that is driven by a part of your small intestine, in particular the portion of the upper part of it that contains this, disaccharidase. Disaccharidase is a class of molecule we call an enzyme. We generally designate enzymes as anything that ends in the letters ACE. Disaccharidase is a class of enzymes that breaks disaccharides like sucrose down into their monosaccharides, glucose and fructose. That occurs pretty much in your small intestine to any great extent. There's some disaccaridases in your mouth and so forth, but most of it occurs in the small intestine, which is why this step in your digestion of sucrose occurs really in the small intestine. Okay, so what is it that disaccharidase does? Well, disaccharidase is an enzyme. And we've already talked about enzymes in lab, but if you haven't gotten to the lab yet, an enzyme has to deal with this problem. An enzyme is something that's going to get us past this problem. If we look at this reaction, let's go back up to this reaction. Here we have sucrose and water, and we take the sucrose and water and we convert it into glucose and fructose, and that has a delta G that's negative. But if you look at the actual energy curve, it looks like this. This is the free energy, and here you've got the reactants. This is the sucrose, so there's the glucose, there's the fructose, and this is the water. This is the OH, and this is the H. In order to get this reaction to occur, I have to break this bond between the glucose and fructose and the bond between the OH and the H in the water, and rearrange them so that this bond now reflects down to here, and you get glucose bound with an OH, and you get fructose bound with an H. When you do that in this configuration, the Gibbs free energy is down here compared to where you started up here. Again, remember, when we calculate delta G, we calculate the free energy at the end minus the free energy at the beginning. We're taking a small number, subtracting a bigger number, and that is going to give us then a negative delta G. So that tells us this is spontaneous. But it doesn't happen on the shelves, and it doesn't happen in your coffee, because there's another part of the story, and that's this part right here. You see this curve? That is representing the energy that's required to get this reaction started. Okay, so in order to get this reaction started, these are in a stable point right here. They're fairly stable chemically and physically. I have to destabilize them by adding this energy. When I get them to destabilize, they talk about the bonds being in a transition state, and that's essentially what's happening, is that the bonds are kind of there, they're kind of not, and then they reform at their lower energy state. Okay, and that's why it is that we end up going down here to glucose and fructose. But this energy that I'm adding, I can add as heat. So for example, I can get this reaction to occur faster if I heat the bag up, or if I heat the water up in my coffee. But even coffee, even though the temperature of coffee is not enough to get this to happen in any appreciable way, I'd have to heat this up a lot more even than what's in coffee. And if you buy coffee, of course, you know that it can scald you when it first comes out. So what I'm saying is you got to get it hotter than that. In order to get this to occur in your body, you would have to heat this up to the point where you yourself would catch on fire. So we can't do that. That's not going to work. So I need this to happen, though. I need to break this down, because if I can't break this down, I can't then take the glucose and fructose and break it down further and release the energy to make ATP. So I've got to come up with a different solution. I can't just add energy to do this. By the way, this energy that's being added is called the activation energy, or the energy of activation. We abbreviate it E sub A. Now, what we're going to do then is this. We're going to come up with a different way of doing this. There's already energy in the system. There's thermal energy in the system. We've talked about that before. So this is bouncing around here, and so it goes up this energy curve a little bit. It just can't get over it very easily. The reason it happens slowly is that occasionally there'll be enough kinetic energy to get it over the energy barrier for one or two molecules, and it'll drop down in here. But there needs to be more energy to get this to happen at any kind of appreciable rate. So instead, what we're going to do is we're going to use not heat, but we're going to use enzymes. By definition, an enzyme is the easiest definition you'll ever get in this class. An enzyme is a protein catalyst. That's all it is. Okay, so it's a protein. You know what a protein is. It's a polymer of amino acids long enough to fold into a tertiary structure. You know that. But what's a catalyst? A catalyst, you've known from your chemistry classes, is anything that changes the rate of a reaction without being altered by the reaction. Now, it's often stated in some textbooks as it increases the rate of a reaction. Most of the catalysts that we know of do exactly that. But the key point is that they're not changed by the reaction. Okay, now we have to interpret that correctly. It doesn't mean that the molecule isn't changed at all. In fact, the molecule can be completely blown apart, can be completely destroyed. But if it's a catalyst, it will be reformed at exactly the same rate. So that makes it a catalyst. Anything that in your reaction doesn't appear to be changing would make it a catalyst if it changes the rate of the reaction. So how is it that enzymes do this? Enzymes do it in a very special way. We talked about this with the activation energy. Here's the activation energy of the initial reaction. In order to get the reaction to occur, I have to bring the energy up to here, and then the reaction will occur, and I'll get the products down here. But the problem is, to get it up there, I'd have to catch myself on fire. But disaccharide lowers this energy of activation that's required to get the system to go. It doesn't completely eliminate it, but it lowers it to the point where thermal energy in the system, your body heat, is sufficient to add the energy of activation to get it over the hump and down into the products. So that's how this thing works. That's how this enzyme works. It lowers this activation energy, allows the thermal energy in the system to get the reaction to go. Okay, now, it's important that you note this. Not all catalysts lower activation energy. Enzymes do that. But in this class in the past, I've noticed when I ask what is a catalyst, a lot of people will answer it lowers activation energy. Enzymes do that. Some other catalysts do that, but a lot of other catalysts don't. So platinum is a catalyst for a number of chemical reactions. It has nothing to do with changing activation energy. We're going to see another catalyst, fumarate, in the Krebs cycle, which also does not change the activation energy at all, but it does act as a catalyst. So be aware of that. We're talking about changing activation energy. We're talking about protein catalysts. We're talking about enzymes, not general catalysts. So as catalysts, enzymes work in a very interesting kind of way physically. The enzyme itself is usually huge compared to the thing that it works on. Here's an example. What you're looking at is an enzyme called hexokinase, and this is not an artist's rendition of the enzyme. This is based on a mathematical model of its tertiary structure. And this is the substrate on which it works, which is glucose. And you'll notice that the enzyme has this kind of mouth shape to it. And there's another binding site over here, and there's some other things that are very interesting that you'll study later in your career, especially if you go into biochemistry. But this site right here is the key site. We call that the catalytic site because that's where catalysis is going to occur. Now, the enzyme has this structure that has a spot in here that fits perfectly this substrate. And it's not just that it fits it physically, but what it does is it also has positive and negative charges, which are opposite the negative and positive charges of the substrate molecule that it's acting on. Now, notice this requires charges, which is why it is that whenever we're looking at something that is living, we expect it to be in a polar solvent because these enzymes require polar bonds in order to function. What happens is that negative and positive pattern inside the mouth will attract the positive and negative pattern. that is on the substrate molecule, such that the substrate molecule will then enter the mouth in a particular configuration. It'll always go in a particular geometry and go in and then interact with this enzyme. Okay, what happens here, what happens next, is fascinating and not very clear and not very intuitive, and we still don't have a particularly good model for what happens in the general cases. We can understand what's happening in specific cases, but general cases is much more difficult. The reason is that what begins to happen here is that the interactions start to occur between the electron shells of the substrate and the enzyme. And those interactions will cause the electrons to start to resonate. So what happens is you have the electrons moving around, and again, if we think of them in terms of being particles, we can think of them as moving and having a negative charge. They're actually wave functions. It's a better way to describe them. But either way, what happens is you get the substrate, which has got its electron moving around. You get the enzyme, which has got its electron moving around, and they get so close together that they start to interact. Their negative charges start to interact with each other, which means the negative of one electron will push the negative of the other. And it'll rebound back because it's being held by the nucleus. So it rebounds back, and then it causes this electron to go this way until eventually they start to resonate. And the electrons start moving together like this. It's a very intuitive and not very precise way of expressing what's actually happening here. Now, the point is, what is happening here is happening at a level of physics that we call quantum mechanics. And the quantum effects are the effects that change the energy of activation required to get that reaction to occur. And again, it has to do with the fact that the resonance pattern that you get in the wave functions is, in a sense, decreasing the order. It's decreasing the disorder, I should say. It's causing it to be more ordered, and that's what changes the energy relationships. And so you get what we call the catalytic cycle. Here's another example. This is disaccharidase, and this is an artist's rendition, just a cartoon. But disaccharidase is like hexokinase. It has this catalytic site. And again, the enzyme is huge compared to the actual substrate. Substrate in this case is sucrose. There's the glucose, there's the fructose, and there's glycosidic linkage. So just like we saw with hexokinase, the sucrose is pulled into or attracted into that catalytic site where something interesting starts happening. You start getting these electron interactions and all these things happen, and it's very hard to describe what happens next, but you can think of it like this. This bond right here is not there and there at the same time. That's the best way to describe it that's without using mathematics. Another way of thinking about it is, think about it as it's flickering on and off. The bond is flickering in and then flickers out and flickers in, flickers out. And the amount of time that it spends being together in the bond versus the amount of time it spends being separated where the bond is broken depends on the thermodynamics. If the thermodynamics, the delta G, is going from this direction to this direction, so from sucrose down to glucose and fructose, then it spends more time with the bond missing than with the bond there. Or, better way of saying it, what happens is this enzyme is then going to open up at some random time. When it opens up, either the bond is not there and you get glucose and fructose, or the bond is there and you get sucrose again. So it can happen that sucrose goes into the catalytic site and then gets spewed back out as sucrose. It can happen that sucrose goes into the catalytic site and gets spewed out as glucose and fructose. Which one is more likely? Is it more likely to be sucrose or is it more likely to be glucose and fructose? That depends on the thermodynamics and the concentrations and all the other physics that are involved. But the thermodynamics is what drives all of this. And this is the key point. Enzymes don't determine what reactions can and cannot occur. All enzymes do is speed the reaction. That's all they do. What determines what reactions do occur spontaneously and what reactions don't is determined entirely by the thermodynamics, by that delta G that we talked about before. So this is the catalytic cycle, and you'll see this in the laboratory. The enzyme then will bind with the substrate. So here's E, that's the enzyme. S is the substrate. When it binds, it forms a complex, and that's the ES complex here. And that's why this arrow is going to the right. Now the complex can fall apart either back into the substrate, in which case it goes back into E and S, or it can release the products, P plus the enzyme. But notice the enzyme goes back to the original configuration that it had before. It does change. If we go back up here, you notice that once that substrate is into the enzyme closes down around it. It's like the mouth sort of closes on it. And that is a change in the structure of the molecule. But then when the substrate is released, the enzyme goes right back to the original configuration. And that's what makes it a catalyst, because it itself is not changing by the reaction in the long run. So that's the catalytic cycle, and that's how enzymes work. Now, what exactly are the enzymes doing? And what is the metabolic process that all of these enzymes are controlling? And how many enzymes does it take to control all those processes? Those are questions we're going to address here very shortly.